thank you for coming to seminar today. Your speaker will be me. So for people that are online, hello, I'm Scott Hamilton. I am a professor here at Moss Landing Marine Laboratories. I run the ichthyology lab, the fish biology lab, and I will be your speaker. A little bit about my background. See, I did my bachelor's degree at Princeton University, and then I went to UC Santa Barbara for my PhD. I refused to leave Santa Barbara, stayed around for another 4 years for a postdoc, and then I started here in Moss Landing. It was 13 years or so ago. And mostly my lab works with fishes, studying what's happening in kelp forests and coral reefs. We do a lot of work related to climate change, fisheries, and marine protected area management and evaluation, as well as aquaculture. And today I'm going to be telling you about some of the work we've been doing, studying the responses of the network of marine protected areas in California that's now been in place for a bit over 10 years, right? And so the program I'm going to be telling you about is the California Collaborative Fisheries Research Program. So you can see it's a large collaboration between a number of different academic institutions. Here's most of the leadership of this group. I'm currently the lead PI statewide for this MPA monitoring program, but it was started back in 2007, really a few years before that, by Rick Starr here at Moss Landing Marine Labs and Dean Wendt at Cal Poly San Luis Obispo. And they really created this wonderful, wonderful program. And maybe it was about 6 years ago or so when there was a big effort to do a lot of large-scale marine protected area monitoring in a whole variety of habitats. Rick was going to be leading the deep water monitoring program. He didn't want to lead two programs, and so he decided to hand me the keys to this program. And I'm just trying to be a good steward of it and make sure I don't crash the car and keep this program going, because it really is fantastic. All right. So here's where we're going. So I'll tell you briefly a little bit about background about CCFRP and the MPAs and the network in California. Jump pretty quickly to the methods that we use to do these surveys to evaluate what's happening, and then really jump into a lot of the results that we've been finding over the past couple years. So we have a long-term time series. It's over 15 years now in Central California, and then the program expanded statewide. Now it's been about 7 years, and so then we have this shorter but much larger geographic scope of surveys. I'll show about 5 years of data here, but we have 7 or 8 now. And then we tag and release fish, and when we recapture them, we get information about their movement. So we'll talk about that and whether there's some evidence of spillover or these fish sort of staying in the boundaries of the MPAs. And then we've done some surveys as well of the recreational anglers, the volunteers that are part of our program, to try to understand kind of their perceptions about MPAs and fisheries management and conservation. So we'll talk about that. All right, so CCFRP is, again, the California Collaborative Fisheries Research Program. It's a community science or citizen science research program. It's a fishery independent catch and release program, so we use standardized hook and line surveys. And the program was developed using the expertise of the fishing community, really from the get-go. Right, these fishermen know a lot about the ocean. All the fish that are caught are from volunteers from the recreational community that come out and fish with us. Some people have been fishing their whole lives. Other people, the first time they catch a fish, they come out with us. All of you in the room could also come out and go fishing with us or serve as science crew on this program. So if you want to go, get in touch with us. Right? And we also utilize the knowledge of the sport fishing captains and their crew to decide where to fish, how to fish. Helped us a lot to decide what sites to use, especially the appropriate reference sites outside of our marine protected areas. Right? We have the academic scientists. Right, so we're involved in running the program and establishing the standardized protocols, collecting the data, dealing with the data, analyzing the results, right? And then resource managers, particularly from California Department of Fish and Wildlife, NOAA, National Marine Fisheries Service, and the Ocean Protection Council, and others are heavily involved as well in the program, and they're the ones that are really utilizing the information to make decisions about how we manage these marine protected areas. Again, sort of the overview of the program, it was designed to conduct scientifically rigorous data collection and analyses, both for evaluating the performance of our marine protected areas, as well as for use in fisheries management. So I won't talk about that today, but the data from this program has been used in 7 to 9 stock assessments of nearshore species. It's really vital information that we're using to understand how these fish stocks are changing along the coast, particularly because the nearshore species that we are working with tend to be what's considered data poor or data limited. So our program's been really helpful for that. And then we also conduct a lot of outreach and education, right, to the angling and community. Right? So why collaborative research? I mean, this was a big emphasis when Rick started the program years ago, and really the goal was to try to engage the fishing community and the scientists, right? Engage them in the science and the co-management of the resource that they're utilizing. At the time, there was a lot of headbutting going on, right? People were coming from different perspectives, and so the goal was to, have the fishermen and the scientists and the managers all working together, collecting data together, right? Creating this shared understanding about the status of the resource, so you're really coming at it from the same direction, right? Really trying to generate, again, this fine-scale spatial data on fish stocks, right? The goal, again, building a lot of trust, right? And facilitating communication among the key stakeholders, scientists and resource managers, with the goal of getting people out there and seeing for themselves how these places are changing. Hopefully that's going to increase some ocean stewardship as well, right? In the recreational fishing community. So briefly about MPAs, right? Marine protected areas have been increasing as a spatial management tool over the past several decades. Here's results from a big synthetic study that at this point it's about 15 years old, but we're seeing the same things now from studies all over the globe where marine protected areas have been put in. Things change often, right? It's not too surprising. You stop fishing in an area, you get more fish, right? It's not really rocket science, but there's a lot of variability, right, in terms of explaining why in some places you don't see a big change, other places you do. But here this plot shows across 120 studies in marine protected areas globally. Often you see big increases in the biomass of species, big increases in their density, their body sizes, and diversity. And many of the metrics we'll talk about today that we'll use as a biomass metric, which really combines information on the density and the sizes of organisms into a single metric. Right, in California, back in 1999, the state passed a law called the MLPA, or the Marine Life Protection Act. Right, and that was part of that law mandated the creation of a network of marine protected areas throughout the state. Most of the goals at the time were kind of conservation, right, protecting biodiversity and ecosystem function, right? But there was some secondary goals of enhancing fishery sustainability, right? Mainly it was from the standpoint of, well, if you're protecting species in these areas, you're not fishing for them, there's sort of an insurance factor where those populations will be sustainable. But as well, we started to learn that fishes in those protected areas could also enhance some of the fisheries outside by getting larger, spilling across the boundary and being caught by the fishery, right, or producing a lot of offspring that then their larvae disperse, right, and can seed those areas that are open to fishing. Right, so the map there you see some red squares and some blue squares and other things. Those are the marine protected areas that are set up along the coast. Currently there's 124 MPAs that cover about 16% of state waters, and the implementation of that network of MPAs happened in kind of a piecemeal process, mainly from 2007 to 2012, but there was an earlier process in the Channel Islands, which are off the coast of Santa Barbara in Southern California. That was the first network of MPAs in California, was put in place in 2003, right? And then there was a statewide process going up and down the coast, starting in Central California in 2007, and went up to the north central coast in 2010, and then down to Southern California, and finally up to Northern California in 2012. In each of those regions, there was a different group of stakeholders that was brought in to help design the network based on guidelines that were provided to them from a science advisory team, right? And the best available science went into developing these guidelines, right? So knowing where the habitat is and where the species are. There were some guidelines in terms of how big these MPAs had to be, so they had to be at least like 5 kilometers along the coast, and that was based on what we knew at the time about the typical home ranges of fishes, so it would contain species kind of within the boundaries. The spacing for those MPAs, they needed to be about 50 kilometers apart or closer along the coast, and that was based on what we knew about larval dispersal distances, so they would be connected, right, through the flow, right, of propagules. And then the total area protected, it was some of the modeling and other studies suggested about 20 to 30% of the coastline was ideal, right? We didn't quite meet that, but 16% is pretty darn good. Right? And then you'll see on the map there's a couple different flavors of these MPAs. So the red ones on the map are state marine reserves. These are fully no take of any organisms. The blue ones that you'll see on there are the next most common. Those are state marine conservation areas, and often those are sort of partially protected reserves. Each one is different in terms of the regulations. Some will maybe still allow you to fish recreationally for fish. Others maybe just for salmon or pelagic fishes. Others maybe you could just harvest kelp. right? And so there's some different regulations involved there. All right, so all the results that I'll be showing you today were really based on this big decadal evaluation and review of the MPA networks, which just concluded about a year ago. And currently, the California Fish and Wildlife and the Fish and Game Commission are receiving petitions from people to, based on the results of lots of different long-term monitoring programs, right, to potentially make changes to the MPAs. They're supposed to be adaptively managed, right? Will things change? We don't know, but we'll find out kind of within the next year. And so it's not just our program that's involved in the long-term monitoring, but there's other programs and other habitats. And another point to make is that Moss Landing is really one of the leaders in the state for monitoring MPAs. So not only are we running this collaborative fisheries program, but Rick was leading the deep water MPA monitoring program that Amanda was also involved with. I'm also involved with monitoring in the surf zone and the sandy beach habitats. Ross and Kevin started with this process an estuary MPA monitoring program. And so Moss Landing is involved with monitoring 4 or 5 out of kind of the 6 habitats that are actually monitored. Right, and so for this big decadal review, it was the first 10 years after the entire network of MPAs was implemented in California. The state wants to know what's happening. Are things changing? Are they working? Are they not? And there was a whole slew, I don't know, there's 60, 80 different questions. Not all of them applied to our program, but I've consolidated those into a few that I'll tell you about today. And so one of the sort of consolidated questions was, are there differences in the abundance, size, structure, and the biomass of fishes inside and outside of MPAs, right? And has that changed over time? Secondly, there's a lot of interest in whether these MPAs are providing any climate resistance or resilience. Third, we'll start to evaluate kind of what factors might be explaining some of the differences in the strength of MPA responses, kind of how well different MPAs are working. Some may be working well, others not so well. And so what may explain that is we've looked at differences in fishing pressure that's happening outside the MPAs, the size, there are different sizes. So the size of the MPA, the area, the age, right? There are different ages now. They're implemented at different times, right? Or the geographic location, right? There's big differences in the fishes in this case between southern and northern California, right? There's big differences in population size. So those factors may also be important. Then we'll use some of the tag recapture data from the program to ask whether there's any evidence for spillover, right? Are fishes staying within the boundaries of the MPAs? Are they crossing over the boundaries, right? Are the MPAs large enough to contain the home ranges of these fishes? And then lastly, how have perceptions, right, of the MPAs changed for volunteer anglers that participate with our program? Do they have more positive views of the MPAs after they come out and fish with us and start to see some of the changes for themselves? So again, here's California, right? Most of our analyses regionally are broken down into these 3 regions. So Northern California, there's 45 MPAs, right? Central California, 29, Southern California, 50 of them, implemented in different times, right? And the program has these 6 academic institutions that are involved in running the program, and each institution has 2 marine protected areas and 2 reference sites that they monitor every year, 3 times a year. And so in the north, we have Cal Poly Humboldt, and then UC Davis Bodega Marine Lab. Central California is us, Cal Poly San Luis Obispo. Southern California, UC Santa Barbara, and Scripps, UC San Diego. Okay, the sampling design, as I mentioned earlier, was developed again from the get-go, involving stakeholders. And so Rick spent a lot of time meeting with fishermen and scientists and resource managers, holding meetings. You can see that there's our seminar room circa, I don't know, what was that, 2005 or something like that probably. The carpets were a little cleaner, I think, back then. 1950, yeah. And so bringing in people, right, with a lot of knowledge on each section of the coastline, helping us to develop these standardized fishing protocols and develop those best practices, and looking at a lot of maps and figuring out, inside the MPAs, like here's the rocky habitat that we think is good, to set up our sampling sites, and particularly outside the MPAs, where's an appropriate reference site that has equivalent habitat so we can compare and contrast changes over time. So the stakeholders were key to that. All right, where do we sample? So here's an example. These are the 2 MPAs that Moss Landing has sampled since 2007. Inyo Nuevo up north, Point Lobos just south of Monterey. And at each sampling site, there are these fixed 500 by 500 meter grid cells, right, that we go and do our fishing in. Right? Here in red, these are the grid cells inside the MPAs. Right? And then here in blue, these are the grid cells outside the MPAs and the reference sites that are still open to fishing. And each day we go out, we randomly select 4 of those cells to go fishing in, and the goal is to fish for 45 minutes inside each one of those cells, trying to do 3 15-minute fishing drifts where we're catching fish. Right? And then, but sometimes the weather doesn't cooperate, sometimes we do more drifts, but the goal is we fish for 45 minutes inside each one of those cells each day we go out. Right? And then lots of information is collected. It's pretty chaotic. We usually have 6 to 10 anglers that are on board catching fish. Right? Each one of them is at their own fishing station with a labeled bin. Right? We're keeping track of the start and stop time of the drift, so the time that we're fishing is a really critical parameter. We take GPS coordinates so we know where or what habitat we're drifting over. In keeping track of each angler, they put their fish in a bucket when they're caught, and we take the fish, grab them, and run, and we identify them and count them, and we measure their lengths. Right? We identify what condition they're in, whether they've suffered any damage from the hooks or fishing, whether they're exhibiting signs of barotrauma, which is pressure-related injuries that fish can experience when they're quickly brought to the surface. If they're in good condition, then we'll tag them with a little external T-bar spaghetti tag so that if they're re-caught again, we can get information about how far they moved and how much they've grown. And then the fish are released. And many times if they're experiencing barotrauma, they're descended. And so we have these descending devices. You can clip onto the fish. Basically, it's like reverse fishing. You'll fish them back down to depth, release them. That recompresses them. They swim away much happier, and they have really high survivorship. That's pretty much the only kind of fishing with hook and line that I'm good at is the reverse fishing. Okay, and so here is the sort of spatial scope, right, of the program. So we have 4 MPAs up in Northern California, right? 4 in Central California, 4 down there in Southern California. You can see the distribution of sampling cells inside the MPAs, and then take a look at the distribution of sampling cells outside of the MPAs right there. You can see they're increasing distances from the reserve boundary. That'll be important later as we start evaluating kind of how the patterns of catch and biomass change as you go from the center of the reserve across the boundary at increasing distances where we're looking for some evidence of fishing the line behavior. Right, here's a summary of the program through 2002. A lot of effort, right, has gone into the 700 sampling days at sea, 31 different boats, 53 captains, over 2,000 individual volunteer anglers have come out fishing with us. And we've caught over 200,000 fishes of 95 species, and tagged and released about 75,000 of them. So really heroic effort, particularly by a lot of the people that are in this room that are involved with the program. Okay, so let's look at some of the results. What have we found, right, from the long-term monitoring? And we're going to focus first on the Central California results for where we have the longest time series. 15 years here is they'll show it's about 17 years total now. Right, so how do we measure the differences in abundance of the fishes inside and outside of the MPAs? Well, we use catch per unit effort as our measure of relative abundance. And so we're keeping track of the number of fish that are caught, right? And you can sum up either for an individual species, the number of individuals, or across many species, right? How many fish are being caught. We divide that then by the amount of time that's spent fishing on each of those fishing drifts to standardize for the amount of effort. Multiplying that by the number of anglers, because sometimes there's more anglers on a boat, sometimes there's fewer. And we keep pretty exact track for each angler how much time they spent fishing. Sometimes your hook gets caught on the bottom, you have to change your gear. Maybe you just decide you want to take a break, right? So we subtract off any time that an angler is not able to be fishing. And then mostly what I'll focus on today is another metric, which is taking that catch data and then adding in estimates of the weights of the fish based on their lengths. There's published length-weight relationships for all the species, so we can get a pretty accurate estimate of their weight. And instead of summing up the numbers, we sum up the weights to get this biomass metric. Biomass per unit effort, or we call it BPUE, which is the kilograms of fish that are caught per angler hour of fishing. And so that's what we'll focus on. All right, so here's the trends then from Central California. Right, over on the left-hand side is the average sort of responses across all the MPAs in Central California. The red dots and lines is the marine protected areas. The blue ones are the reference sites. Those colors are consistent throughout the rest of the talk. Over there on the right-hand side then are the patterns for the 4 MPAs that we survey, so you can see individually what's happening. So if we start overall, right, the general trend is the MPAs and reference sites were pretty similar in fish biomass when the MPAs were first implemented in 2007. They didn't really differ that much for the first 7 years, and then you'll see the biomass in the MPAs really took off. And in general, there's about twice the biomass of fishes now inside the MPAs than there are outside. So big increases, especially recently. The other important point is that you look at the reference sites, right? I mean, they're both going up, right? Fish biomass is going up both in the MPAs and in the reference sites, which is good. There was a lot of concern early on that fishermen would just redirect all their fishing effort outside the MPAs and just fish those populations down. So we're not necessarily seeing that. And so likely there's some beneficial environmental conditions, right, as well as some potential benefits from the MPAs actually enhancing those areas outside. If you look at the 4 individual locations, you'll see there's some pretty different responses, right, that are happening. Point Lobos and Point Bouchon down off Morro Bay, big increases in biomass, right, inside the MPAs, and not much of a change in the reference sites. Contrast of Piedras Blancas, it's going up both inside and outside the MPAs, but faster inside the MPAs. And in Nuevo, you'll see there's not much of an effect of the MPAs at all. Biomass is going up, a little bit, both in and out. So we'll come back later to what may be explaining some of those differences. If you look at some of the individual species, right, that we're catching, you'll see there's some different patterns. In general, all these species, there's higher biomass inside the reserves, and overall 73% of the species had higher biomass inside the MPA, so it's not just one species driving the patterns. And some of the things you might notice are some of these species here: blue rockfish, olive rockfish, vermilions, coppers, gophers. Right? They tend to show a pattern where the biomass inside the MPAs has really increased in the last 5 or so years. Right? So you see those big differences kind of in the recent parts of the time series. Whereas there's other species here, kelp rockfish, particularly you see it well in lingcod, canary rockfish, yellowtail rockfish. Still, again, there's higher biomass inside the reserves, but that biomass really peaked in about 2015 and has since declined. So we'll talk about that in a little bit as well. And then there's other species here like black rockfish, which are actually more abundant outside the MPAs than inside. And you'll see an explanation for this later as well, and particularly this is influenced by what's happening at the Año Nuevo MPA. Okay, what can the program show about, or say about climate resilience and resistance of the MPAs? So the long-term time series, right, crossed a large marine heat wave, which you are all probably familiar with, right? The big warm blob event that happened from 2014 to 2015, and the El Nino that happened after that in 2016. There was a period of 3 years where the water was much warmer than normal. And so if you look at those plots there, this shows sea surface temperature, right, the differences from the long-term average. And the blue and yellow colors that you see in 2012 is when ocean temperatures were about average. You go to the far right, 2018, after the heat wave, again, those blue to yellow colors, temperatures were about average. But then you look during the middle of the heat wave in 2015, everything's red, right? The ocean temperatures are about 3 degrees warmer, right, on average, 3 degrees Celsius warmer on average during that period. All right, so what did we see in terms of the responses of the fishes? So now the metric that we're looking at is a measure of biodiversity. So the diversity in the system, the Shannon Diversity Index. You'll see at the beginning of the time series, diversity was pretty similar inside and outside of the MPAs in Central California. And then diversity increased inside the MPAs. It was higher, right, than those sites open to fishing. In gray there is when the marine heat wave happened. And you'll see kind of right after the marine heat wave, diversity collapsed, both inside and outside, right, of the MPAs. And so the MPAs didn't really do a good job resisting the heat wave. But then if you look after the heat wave, you'll see diversity increased more quickly inside the marine protected areas than outside. So this is some suggestion there may be a bit more resilience of these marine protected areas to those climate disturbances. And what was leading to these changes in diversity was really some changes in the species composition of the fishes that we are catching. So you'll see the MPA sites there on the left, the reference sites on the right, and then it's organized before the heat wave, during the heat wave, and after the heat wave for each one. And so I'll highlight some of the changes in the fish communities that we saw. So in particular, here's black rockfish. You'll see both in and out of the MPAs, they became a much smaller proportion of the community following the heat wave. Whereas a species here like blue rockfish really increased, right, following the heat wave and became a much larger proportion of the community. In particular, a lot of the planktivorous fishes really became to dominate the community. Right. We also looked in relation to some climate indicators and climate indices. So the multivariate oceanographic climate index here, MOCHI, takes information about upwelling and productivity and kind of puts it into one metric. And what we found is that the diversity of the fish community is strongly related to the oceanographic conditions with a 2-year lag, right? Because it takes a while. This is likely influencing fish recruitment patterns. We don't start catching the fish with hook and line until they're probably about 2 years old, so there's a bit of a lag until we see this effect. But when this index is very positive, you'll see diversity is low, and that's reflecting periods that are these warmer waters, less productive, kind of El Nino type conditions, right? Whereas diversity is much higher when the waters are cooler, right? More productive, more like La Nina type conditions. That's a more negative value of that index. So the oceanography, again, can be really important in influencing these fish populations. Right, here's the same plot for all the top species that I showed you earlier, but now we're overlaying the timing of the heat wave, so you can see where that gray bar comes down. And again, some of the species, like we talked about earlier, blue rockfish, olives, coppers, gophers, those ones are species that really increased after the heat wave. They likely had really strong recruitment in the years sort of right around the heat wave. Whereas like lingcod and canary rockfish that we talked about, you see their populations really peaked during the heat wave, right, and then since declined. Right? So these climatic disturbances are individually having different impacts, right, on the different species that we're studying. So now I'll tell you about another way we've utilized the data in a different metric, which is kind of looking at the effect size, or it's a response ratio. It's typically used in the meta-analysis literature. And it's a way we can synthesize the data and compare sort of the magnitude of effect that we're seeing. And so here we take the log of the biomass inside the MPA divided by the biomass in the reference site. The log of 1 is 0, so if the biomass is equivalent in and out of the reference sites, you'll have a value of 0. If the biomass is higher inside the MPA than the reference sites, it'll be a positive value. The more positive it is, the bigger the difference in biomass between the MPA and the reference sites. If this value is negative, then there's lower biomass inside the MPA compared to the reference sites. So now let's look at the same data from Central California, right, over time, but now in terms of this response ratio metric. Right, and so again, on the left is sort of the average response. On the right is the individual responses of those 4 MPAs. And so you'll notice at the beginning of the time series, right, that response ratio was pretty close to 0, right? There was pretty similar biomass inside and outside of the MPAs. And then over time, right, this response is increasing, showing us that the difference in biomass between the MPAs and reference sites, right, they're diverging in biomass over time. Biomass is building up in the MPAs, and it wasn't really changing much outside. So that MPA response, that effect, right, is increasing over time. If we look at our 4 individual MPAs, again, you'll see very different responses. Point Lobos and Point Bouchon, you see these really strong responses, right? Biomass keeps building up over time in the MPAs. And as we saw earlier, it wasn't really changing outside. Whereas if you look at Piedras Blancas and Año Nuevo, again, there's not much of an impact. It's still positive, but you kind of got to squint to see it. So again, what's explaining the differences in response of these different MPAs? And so we looked at a whole variety of things: oceanographic conditions, temperature, wave exposure, productivity. What really jumped out as being an important factor that could explain those spatial differences and the strength of the MPA responses was the amount of fishing pressure and fishing effort that was happening in those reference sites outside of the MPAs. So here's an example: Point Bouchon, MPA off of Morro Bay. Right, in red there, again, these are the cells where we're sampling inside the MPA. Right, the blue cells there, that's where we're sampling in the reference sites, and then overlaid on top of that in gray is from the California Department of Fish and Wildlife, what they call their fishery microblocks. So 1 by 1 nautical mile blocks, and they do surveys of the recreational fishing community and ask them, like, where do you go fishing? And so we got access to that data and could then build our own index of fishing effort in those microblocks that overlap with the reference sites where we're doing the sampling for CCFRP. And could that way come up with an index of how much fishing pressure is happening outside each of these MPAs. So here's what that looks like. Right, so our fishing effort metric is on the x-axis. That MPA response ratio is on the y-axis. And you'll see there's this positive response. Right? the more fishing effort there is, the bigger the difference in biomass inside and outside of the MPAs. So over there on the left, right, where you're not fishing, where there's not much fishing effort outside the MPAs, right, the fish biomass is pretty similar in and out, which you would expect. If you're not really fishing there, right, they should track each other, right? But in places where there's really heavy fishing pressure, there's a lot more fish inside the MPAs and not very much outside. And if we look at our sites, right, here's Año Nuevo and Piedras Blancas, right? The two sites where we saw a pretty weak effect are the sites where there's not much fishing happening outside. These places are much further from port, they're harder to access, so that makes sense. Right, and up here, this is Point Bouchon and Point Lobos. Again, the sites where there's much heavier fishing pressure outside are the sites where we're seeing these strong differences inside and outside of the MPAs. Again, closer to port, much heavier fishing pressure, so that makes sense to us. Okay, so let's shift gears and let's look at the statewide program. So now expanding beyond Central California to add in Northern and Southern California. So it's a shorter time series. Many of these sites, we didn't start sampling them until after the MPAs were established, right? But if we look at some of the spatial patterns, here after about 5 years of sampling, all the sites are arranged from north to south, and they're paired in red, the MPAs, right? In blue, the biomass and the reference sites. And if you scan all the way down, you'll see in every case the biomass is higher inside the MPAs than the reference sites. So that's a good sign, right? These MPAs are working pretty much everywhere, right, across the state. But you also notice in some places there's a really big difference in biomass inside and outside of the MPAs, like Stewart's Point up in Northern California, or Point Lobos, right, in our backyard. Other places, there's not much of a difference at all. Cape Mendocino up north, right, Año Nuevo in our backyard. So again, the question is, what factors are responsible for some of these spatial differences, right, in MPA effectiveness? So that's what we started to investigate here, right? So here again, looking at some of these response ratios, this just shows you over time the response ratios at all of those sites. It's kind of a big pile of spaghetti, right? But the things to really take home are that almost all the points are above zero, right? So there's more fish inside the MPAs than outside across all these sites up and down the coast. And in general, you might also see a bit of a trend where if some of the warmer colors are up top, the cooler colors down low, which you'll see in a second in terms of there's a bit of a latitudinal effect on the strength of the MPA responses that we're seeing. Right, so the factors that came out in this analysis to be really important in terms of explaining the differences in MPA effectiveness were the size of the MPA. Right, so again, on the y-axis, that's our response ratio. Right, the x-axis on that first one is the total protected area of the MPAs, and you'll see larger MPAs, right, ones that are bigger, there's a stronger response ratio. There's a bigger difference in and outside of the MPAs. Right, smaller MPAs, they're much more similar in biomass in and out. Right, secondly, the age of the MPAs are very important. Right, so how long they've been protected, the longer they've been protected, the more time for biomass to build up. And as well there, we see this positive response, right? The longer the MPAs have been protected, right, the bigger the differences. And then lastly, location on the coast was also important. So latitude, right, from Southern California on the left to Northern California on the right. And there you see the opposite response, right? So typically we're seeing stronger MPA responses in Southern California than Northern California, which I think can be explained by the fact that there's a lot more people in Southern California, there's a lot more people fishing there, right? It's also the waters are warmer, the fish that are there, they grow faster, so you would expect them to respond faster. And typically those species tend to have sort of more consistent recruitment and replenishment of the populations. In contrast, in Northern California, there's a lot fewer people, there's not as much fishing that's happening out there, the waters are cold, fish grow really slow, so you expect it to take longer for those differences to develop. And the fish populations tend to be replenished much more episodically, maybe one or two good years in every 10, so it takes longer. And then still, if we look across all the different species, like don't worry too much about what species is which, just look at the patterns from Northern California up top, Central California, and Southern California. Again, 0 is where there's no difference. You'll see almost all of the bars are on the positive side, right? Suggesting that for the vast majority of species, they have higher biomass inside the MPAs than out in all of these 3 bioregions along the coast. Right, there's a few species that show the opposite response. Often those were species that we encounter much less frequently than some of the others. So again, across all these regions, over 70% of the fish, there's more biomass inside the MPAs. So again, good signs that they're working kind of as intended. Right, so now we're going to shift and talk about the tag recapture results right? Asking, is there any evidence for spillover that's happening, right? Are fish staying within the boundaries of the MPAs, right? Or are they crossing the boundary? They don't know where the boundary are. There's no sign for them to tell them to stop. And so the question is, is this happening, right? And kind of what's the rate at which fish may be spilling over? So again, another part of the program is these tagging and recaptures. And when the fish are recaptured, we get information about where they've gone. So each fish is tagged. We use a tagging gun, put in these, one of these little T-bar tags, and they can stay for years in the fish. So on the bottom, you can maybe barely see one of these tags and see how fouled it is with algae. So sometimes, you'll catch a fish and it takes a while to realize, oh, it actually has the tag in it. Right, so when we release the fish, we get information on their location and all that, and we put up these flyers, in different harbors and send them to different fishing clubs to try to, tell people that are out there fishing for themselves, if you catch one of these fish, we are very interested in it, and we'll give you a reward. So there can be a cash reward or some swag from the programs and stuff like that. And we try to get, we give people photos that here's what the tags might look like if they're really fouled, and we ask them to record information from us in terms of how big was the fish, where did you catch it, what depth was it at, those sort of things. And then for people that report those catches to us, then we'll send them an informational flyer about their fish. And we'll tell them, yep, here's where we caught the fish, here's where we released it, here's where you caught the fish. We'll calculate how long was it out there before it was caught again. Here's how far it swam. And then as well try to provide some information on the natural history and life history of that species that may be of interest to them. All right, here's what some of the results look like. Again, we've tagged tens of thousands of fish, but the recapture rates are pretty low. They're less than 1%. Inside the MPAs, we're really the only group that's out there fishing, and we go 3 days a year. Outside the MPAs, there's more of a chance they might be caught. Right? But what we can find from the couple hundred of recaptures that we've had over the years, right, on the left there, that shows you a histogram of all the recaptures, sort of the net movements. How far did the fish go from where we released it to where we recaptured it? It could have traveled a lot of other places. We don't know. All we know is here's where we released it, here's where we recaptured it. But in general, you'll see the fish, they're not moving very far. Right? The bulk of the recaptures are in less than 1 kilometer from where the fish was released, occasionally a bit more. Here's an example of gopher rockfish that tend to mainly fall on this side of the histogram with very low rates of movement, typically less than a quarter kilometer from where they're released. And so here's an example over there, that map from Point Lobos. You can see some of the arrows showing cells where they were released and then where they were recaptured. Right? Some are moving in, some are moving out, but the bulk of the fish are caught often in the same cell where they're released. They're not very mobile. Here's another example. This is copper rockfish. Right? They show a bit of a different pattern. It's kind of a bimodal distribution. The bulk of the fish don't move. 3-4 years later, we'll catch the same copper rockfish in pretty much the exact same hole, right, where we caught them and released them originally. But there's some fish that will make these longer movements of 10 to 100 kilometers, and so you'll see a few of those sort of occasional long-distance movements that we've seen for that species. And then here's a species on the opposite end of the spectrum. These are black rockfish. They tend to book it. And so they're mainly in this 10 to over 100 kilometers of movements when they're recaptured. Most of the fish that we're catching are immature in size, and they almost always swim up to Northern California and Oregon, and we think probably to spawn, right, and to mate. But it's pretty much, except for one fish that's always been unidirectional, right, up to the north. Right, and so then we can also try to assess kind of what's the rate of spillover that we can calculate with our data using the fishes that were tagged and released inside the MPAs. We can ask what proportion stayed in the MPA or crossed the border and were caught outside. So we've tagged 25,000 fish in the MPAs, and we've recaptured 136. So a pretty low recapture rate, but again, the effort isn't as high as you would want it to be necessarily to really do a good tag recapture study. But from those recaptures, we find that 17% of the recaptured fishes spilled over. They were caught. They were tagged in the MPA, and they were caught outside. So is 17% the true spillover rate? Who the hell knows? Probably not, right? But at least it's giving us, a window into what it may be. It's not zero, right? Some of the fish are certainly leaving the MPAs. It's not 100%, right? A lot of them are staying inside the MPAs, right? But we're getting a sense for how this is. And if we look across those 4 Central Coast MPAs where we have the most data, you'll see there's some variability in the amount of spillover. So in blue, those are the proportion of fish that stayed in the MPA, right? So again, the vast majority did. In red are the ones that
left the MPAs and were caught outside. And you'll see anywhere from 5 to 25% of the fish in any of these sites cross the boundary, right, and spilled over. Another thing to point out, those two sites where we saw the weakest MPA responses, Año Nuevo and Piedras Blancas, are also the two sites where we have the greatest amount of spillover that's happening. So that may be another piece of the puzzle, right, for why those reserves are not as effective. In Año Nuevo, mainly that's these black rock fish that are taking off, right, and moving up north, whereas at Piedras Blancas, it was mostly gopher rockfish. They weren't moving very far, but they were just kind of moving just across the boundary, not knowing where it is. All right, sort of the last bit before we talk about the survey results is looking at some evidence for fishing the line behavior given our data. This is work that Sal Jorgensen was doing, and current postdoc Nate Spindle is really helping out with a lot of the analyses on. And so this is asking, like, what happens when you look from the center of the MPA to the boundary and an increasing distance further from the boundary? What sort of patterns do we see? And here's some conceptual diagrams of what it might look like. Right, the top one on the left could be, if there's sort of just heavy fishing pressure everywhere outside the MPA, you would expect high biomass inside, right, or catch rates, and then low outside. If spillover is happening, right, densities build up, it gets crowded, fish start leaving the boundary, you might expect to find higher catches just outside the boundary, and then it would decline. On the bottom there, panel C, that's what we would expect if fishing the line behavior is happening, and in many places we know fishermen like to target the boundary of the MPA. They know there's big fish inside. They'll occasionally cross the boundary. And if you have the highest effort right at the boundary, you actually may get the lowest catch rates sort of just outside, and then it goes up again. Or there could be what's called these edge effects, right? Whereas you're catching these fish that are spilling over, you may actually depress the density, just inside the MPA, just past the boundary. Or it could be some combination of these things, right? So we use that framework then to evaluate what was happening with our data by looking at, again, the catch rates and the biomass in each of those cells, right, in relation to how far that cell was from the nearest MPA boundary. So the plots will kind of look like this. Right. So up on top is the raw biomass data. Down below, we've standardized it, the biomass for each MPA and reference site pair, because we know there's a lot of geographic differences in the catch rates. So that helps to pull out kind of the underlying pattern that's happening. Right? In both of those cases, you'll see the highest biomass is in the center of the MPA, and you get to the dashed line, that's the boundary. And then it declines as you go across the boundary. You'll see that dip, right? There's that prominent dip a few kilometers past the boundary, which is indicative of that fishing the line behavior, and then it increases again. So that, when we first saw this, this like totally blew me away. It's like, I've never had data that fit like that theoretical pattern, so well. It's like, what did we do wrong? But in Central California, then we can see how does that pattern develop over time since we have that long time series. So I've broken it down into kind of the first 4 or 5 years, the next 4 or 5, the last 4 or 5, and you'll see over time, right, that pattern has really builds, right? The differences in and out really increase, and you see that dip become more and more prominent over time. Right? Nate and Sal have also been doing a lot looking at how fish mobility and fish movements may influence the strength of this pattern, these edge effects that we see as you go further and further from the MPA boundary. And so they went into the literature and categorized the fish based on what we know about their home ranges as to being low, medium, or high mobility classes. And you'll see there on the left, the catch per unit effort, on the right, that biomass metric. Again, you see these really strong edge effects and declines right from the center across the boundary for those low mobility species. Right? And you don't see this very much at all for those high mobility species that are moving back and forth quite a lot. Right? You wouldn't expect to see it as much for those species, and we don't. So really kind of how much the fish are moving and how much they're spilling over the boundary influences sort of the strength of this fishing response that we're seeing. Right, those signals of spillover also, we see that in the size structure of the fish. So now, because we're catching lots of species that, from little ones to big ones, right, the species differ in their body size. So here we've standardized the lengths of all the fish based on their own size at maturation. And so in this case, a value of 1, that's the size at which fish mature. So if the point's above the line, those fish on average are bigger than the size at maturation. If it's below the line, they're smaller than the size at maturation. And here you'll see, right, in general, from the center of the MPA, right, the fish tend to be larger than the size at maturation, right? And then as you go across the boundary, that declines. By the time you get to 5 or 7 kilometers past the boundary, now most of the fish are smaller than the size at maturity. Right, but the interesting thing is, right, that sort of spillover effect goes a bit further than the distance that you saw for the fishing the line effect. So you're still catching large fish, 1, 2, 3, 4 kilometers, right, past the boundary. So this maybe explains, like, as a fisherman, why would you go keep fishing near the boundary if you're catching fewer fish? Well, likely because you're still catching these bigger fish, right, which are much more valuable. If you break that down into these large and small MPAs, you'll see that the MPA size also modifies this pattern, right? Whereas the large MPAs is what we saw before. If you look at the small MPAs, right, that decline in size happens much more quickly, right, as you go across the boundary. So again, indicating how, the larger the protective area, the more effective it is in holding larger populations, sort of containing their home ranges, and probably providing individuals that can spill over, more continually spill over and be caught by the fishery. All right, last part of the talk. This is then about the angler surveys that we did. So some surveys that we conducted with the volunteers that come out with the program, asking them about their opinions on MPAs, fisheries conservation, right, fisheries management. And so we sent this survey out in 2021 to, at that point, about 1,400 anglers that we still had their contact information for. Right, we got about 260 responses, about 19%, which is pretty good for most surveys. And you'll see then the map along the coast, kind of the counties where we got responses from. So, from northern to southern California, typically coastal counties, that's where people come out fishing with us, but not always. Right, and here's a few of the key results, right? So we asked people, like, what were your opinions of the MPAs before you came out with our program? And then what were your opinions after you spent some time fishing with us? Right, so over there on the left, right, shows the change in opinion from before they came out to the program and after. And you'll see, the big obvious pattern is there's a big increase in highly positive views of the MPAs after people came out and worked with the program, mainly because they changed their opinion from these more negative views. Over there on the right, we've broken it down into responses from northern, central, and southern California. Right, in gray up top in the pie charts, those are the people that didn't change their opinion of an MPA. So the vast majority didn't really change their opinion, but if they did, you'll see it's mostly the blue, right, not that sort of beige color, right, and that's a positive change. So people shifted their opinion and often became more positive, not less positive. Down below shows the magnitude of change, like how many steps did their opinion increase or decrease. Again, you'll see almost everyone, if they changed their opinion, it went more positive. Again, the vast majority of the people along the zero line, they didn't change their opinion, but also those are people that already started with a positive view of MPAs. So if they did change their opinion, right, it was because they started with a more negative view. And you'll also notice in Southern California, more often people started with more negative views, right, of the MPAs. And potentially that's because there's much higher fishing pressure down there. The reefs were potentially a bit more degraded, and there was a lot of opposition to the MPAs down in Southern California. But still, those people, after they came out, right, changed their opinion. The amount of time that people come out and participate with the program also has an influence in kind of how positive their views become of the marine protected areas. Right, so over on the left is people that come out like year after year. In the middle, they come out every so often. Right, and then over there on the right, they've maybe just come out once, right, or twice. And so the people that are coming out every year, you'll see there's a much bigger positive change in their opinions of the MPAs. Right? And I think what's happening here is that, over time, you're seeing for yourself, right, the changes that are happening. So we've also asked people, if you fish both in and out of an MPA, where are you catching more fish? Where are you catching a greater diversity of species? Where are you catching larger sizes? And overwhelmingly, people say inside the MPAs, right? And so, scientists like me can come up and show you data and tell you, oh, look at all these changes, right? But people are really going to start believing it, when they're seeing it for themselves. They see they're catching more fish and bigger fish. They go tell their friends, right? I think that's how we combat some of the skepticism about these protected areas. All right, so a little review. Hopefully I've been able to convince you that the MPAs are working pretty well, right, across the statewide network. The fish are bigger, right? They're more abundant. There's more biomass in almost all the MPAs we're sampling. And on the climate side, we didn't see, the MPAs didn't really resist the effects of the heat wave, right, which depressed diversity and resulted in these community changes both in and outside of the MPAs, right, but they did appear to be more resilient, right? They bounced back more quickly in terms of their diversity. Again, Things that were really important in explaining the differences in MPA responses were the amount of fishing pressure that's happening outside, and the size of those MPAs, how long they've been protected for, and their location on the coast. Right, we did see some evidence from the tag recapture data of spillover, right? But in general, the majority of the fishes are staying inside the MPAs, right, for extensive periods. So it suggests they were appropriately sized, right, to kind of protect, right, these populations by encompassing their home ranges. Right, and then lastly, hopefully, we have shown you the sort of the power of this collaborative research. Right, we're doing these rigorous evaluations that are really useful for management, but also the outreach and education is starting to change minds, right, particularly in part of the community that was really vocally opposed, right, to the creation of these protected areas at the outset. So with that, I don't do social media, but many of you do, so you can find us there if you want. And yep, thank you to everyone who came today and to all our partners and all the science crew and the volunteers and the captains and everyone who makes this program happen. Thank you. To entice you to ask questions, Jake gave me some stickers from CCFRP. So if you ask a question, you get a sticker. Questions? Petra. Oh, wait. What do I got to do? I got to talk into this one? Oh, Petra. Okay.
Good. Okay. Thanks. First of all, I'm really glad to hear about this. I mean, I've heard about it before being here, but it's really nice to see it all presented as like an amazing, amazing data set. I was wondering about the biodiversity, and because you showed the diversity for the Central Coast places, and you know, it's obviously really marked with that heat wave. Mm-hmm. Was there a shift in the diversity in like the northern sites? Like, did different species benefit in the northern sites? Like, was the pattern the same across the coast?
Unfortunately, we don't know, because we didn't start sampling with this program, Northern and Southern California, until 2017. So that was just after the heat wave. So we really only understand what's happening in response to the heat wave in Central California. Yeah. I mean, there definitely were some species that have like more southern affinities that kind of moved up the coast, so we started seeing more of these warm water species kind of showing up and sticking around. We've seen that even in Central California, but also up north. So there was some changes that way, right? But yeah, we'd have to look at probably more like the kelp forest scuba program as a way to address kind of how the northern sites, responded to the heat wave. But yeah, good question though. You get a sticker. Duncan's doing it for the sticker.
Oh yeah, of course. So I have a lot of questions, but in the interest of time, is there a lot of evidence of poaching in the MPAs? Is there, because obviously if you've got a big area with a bunch of big fish in it, someone's going to want to go get those fish. So is there a lot of evidence of poaching, or is the enforcement pretty good and people are pretty respectful of it, or is that an impact?
So in California, I mean, there is some poaching, but it's pretty minimal kind of as far as we can tell. I mean, Fish and Wildlife is out there and they're, they have the wardens out and they're looking for that, but it's a large coastline, so it's hard to monitor. Occasionally you'll see people fishing inside the MPAs. Often it's not very often like commercial guys, because they know and they don't want to risk it. It's often recreational anglers. In many cases, they just don't know where the boundaries are. So you go tell them, right? They'll often say, "Oh, thanks for the information," and leave. So there's a little bit of it, but compared to other places where kind of the amount of compliance seems to be a really important factor in determining the effectiveness of the MPAs, here I don't know how important yet it is. In some of the MPAs maybe, but there's a lot of eyeballs out there in many of these places, and so I haven't seen a ton of poaching. I don't know, Rick, would you agree with that assessment?
Yeah, I would just say that the Department of Fish and Wildlife has a limited budget for enforcement, but they do as good a job as they can on that. But what we've seen throughout the state is that the really key part of enforcement are private citizens. And I know a lot of the charter boats who've worked on CCFRP will go in and let people know they're fishing in the MPA, and I think that's been more valuable, or as valuable as the official enforcement.
And there's also another citizen science program on the coast called MPA Watch that has people that go to, not all the MPAs, but a decent number of them, especially ones that are visited pretty regularly, and they're out there recording information about compliance and things, and giving people information so that they know. So luckily we have some of that happening too.
I haven't asked a question yet.
It's going to be a good one, I'm sure.
Thank you. Well, I was noticing how some of the reference sites are located in different latitudinal location in relation to the MPAs. Like, so Año Nuevo, the reference sites to the north, and Point Lobos to the south. And so I'm thinking about, currents and, larval dispersal. So how is it? I was surprised actually that the design didn't include, both sides of an MPA, but just one side. Is that possibly an explanation for some of the?
Often the biggest factor that was used in deciding where to put the reference sites was where was the habitat that was most comparable. And sometimes, like to Año Nuevo to the south, there wasn't really good rocky habitat.
Right.
So it all had to be to the north.
Right.
And so in cases where we could split it on either side, there was equivalent habitat, we would. And Point Lobos actually has sites north and south. But other times it mainly was in one direction, depending where, especially in working with the fishermen and their knowledge, like where, and habitat maps, like where.
Right.
Where's
So
the
like
most equivalent
Rocky
habitat?
South
'Cause we
Strait.
thought that
Yeah.
was probably the most. That was what we had to control for most. I mean, certainly currents. If you're one side of a headland or another, that can influence things. But we tried to most control for those habitat differences, as we thought that would be really important.
So, but there's no relationship between. I mean, I understand that, and that makes sense, right? You're looking for mostly seafloor maps, but
Yeah, where's their good rock?
where's good? I love hearing that. But in terms of dispersal, like,
Yeah.
because obviously, when something's to the north, is going to be a very different.
Yeah.
Is there any relationship in terms of explaining some of the variance in your data set? Or?
We've started to work a bit. So Pete Raimondi at UC Santa Cruz, Mark Carr, their group, they've been doing a lot of, working with Chris Edwards, have been doing a lot of dispersal modeling, right, using ROMs, regional oceanographic models, and looking at connectivity between the MPAs. And so we're just starting to get kind of these metrics of connectivity as a potential response, and we use that in this and some other analyses. It isn't turning out to be like the strongest predictor of sort of the differences in responses that we're seeing, but there's a little bit of an effect in there. So, but it's something we've been worrying about, or not worrying about, but thinking about, and and we're just now starting to be able to actually utilize it. So there'll probably be more analyses we'll do that'll include that in the future.
All right.
Yeah.
Thanks.
Yeah, because certainly if you're putting your your reference site or whatever in a sink that's not getting any larvae, like the population's not going to grow, right? So that's an important consideration. Yeah.
Someone want a sticker first?
Just because I've caught a few. I noticed the lingcod was pretty high during the heat wave, and it seems like it's a kind of an alpha predator, especially in that species, or in rockfish in general.
Yeah. Lingcod are one of the biggest predators in the system. Yeah.
And so I just was wondering if that was your take. Is it because it was just taking advantage of the situation? Maybe there were a bunch of sick or weak fish or whatever, and it was just having a field day.
So I think with lingcod, what explains that pattern, like why they really peaked in 2015 and then declined, I think it might have to do with some of the sort of dynamics with the population replenishment that happens. So maybe every 10 years or so, lingcod will have a really strong recruitment event, and they had a really strong recruitment event, a number of years earlier when we had some really cold, productive La Nina conditions. Like we went out trawling with a marine ecology class, and they were just catching tons of baby lingcod. So there's this really big influx into the population that doesn't happen that often. They then moved from the kind of the sand onto the reef. So they were there, we were catching a bunch of them. Right? Then they will start to migrate as they get older into deeper water. So part of the decline is this sort of ontogenetic migration that happens. It's probably what we also saw with area rockfish. But also they're not very long lived. Lingcod only live like 10 to 12 years. So that was probably one big cohort coming through, right, as well, and then started declining. Okay. The warm water maybe or maybe didn't help after that. And then some of these ontogenetic movements, yeah.
Okay. Yeah. So you think it would just happen no matter what?
I think with their populations, you're always going to see kind of these increases, declines as these cohorts move through and they, and they die and they move offshore and all that sort of stuff.
Okay.
I thought the spillover measurements were really interesting and cool, and I wanted to know if you thought the reverse of that was significant at all, if you think there's a substantial amount of fish coming into
Spill in, yeah. Is there
Yeah.
spill in happening? We looked at that, and there were very few cases of fish that were tagged outside the MPA that then moved in. And it's probably happening, right? Again, the fish don't know the boundaries are there. But as we're the only ones that are fishing inside, probably the chances of catching them are also fairly low. But yeah, there was much more, at least in our data set, there were many more examples of fish spilling out than spilling in, which is interesting too. Yeah. One or two, yeah, it was very, very limited. Yeah, I was kind of surprised. Yeah.
Ooh, I have a question. So typically when you look at these MPA studies, thank you, you'll also see graphs showing how the distribution has changed like throughout the trophic levels. And typically you'll see like as you have an MPA, like the trophic level of different like individuals or different species will increase, so you'll have like more like predatory larger fish. And so I'm curious if you guys have data that shows that within these MPAs, or do you feel like because you're fishing like with lures typically and you're line fishing, you're typically catching like rockfish and more predatory fish, if you feel like your methods are limited in allowing you to see the difference in trophic levels that you might see with other studies that have done similar things?
Yeah, I think a lot of that is playing a part. So, when I did some work in kelp forest where we're looking at the whole fish community, you often see that it's the predators, the piscivores, the carnivores that increase first in biomass because those are the things we tend to fish for, right? So it's often, we're fishing for, we're targeting species that tend to be higher, in the food web, right? So they tend to respond more quickly. And so I think with CCFRP, since we're only catching the targeted species, it's harder to really see those trophic changes as opposed to if we were doing a dive survey or had a video survey that was seeing kind of the whole community, including the, those non-fish species, which tend to be lower on the food web and tend to be the prey. Mm-hmm. We have done some analyses looking at kind of the trophic changes in the system, especially in response to the heat wave, and so there was some trophic reorganization. And often what we saw is that we actually lost some of the piscivorous species and we really gained more of the planktivores, right, the things that were eating stuff out of the water column. So that wasn't necessarily the direction we would expect, right? It was kind of opposite of that, right? But showing how those climate disturbances can have different effects at different levels of the food web.
Cool, thank you.
I'll be fast. Thank you. I learned a lot, and you may have said it at the beginning, but I was interested in the biomass per unit efforts and how those are increasing. Was the catch increasing at the same rate? Can you decouple whether there were larger fish, more fish, or kind of what the combination of changes were that were happening inside of the MPAs?
Yeah, it was both. So the increases in biomass were due both to increases in abundance from the CPUE, the catch rates, as well as the sizes. So when we break it down and just look at the catch per unit effort responses, or if we just look at the changes in sort of the average size of the fish, we see both tend to be going up, right? And then when you put them together, you see an even bigger response with the biomass. And so that's kind of why we focused on that metric, because it combines the two and it magnifies the effect. But generally, if you look at just like the CPU patterns there, the overall trends are pretty much the same. So it's that abundance changing plus some added sizes on top of that.
Cool. Wow. That's great.
Thanks. You didn't talk, thank you. I wondered what the depth range is and some of the criteria for the site selection early on. And this question is related to what the predictions will be for kelp disappearance. And so since that's restricted in depth, some of the kelp, so the deep water kelps might be affected too. But can you talk about what you think the last three, because these plots ended in 2020, and now with wide-scale California depression of kelp, what you think the data will look like for these last 3 years?
So typically the cells for the program, the depth range is about 20 to 40 meters typically. And so we limit the depth so that we limit the effect of barotrauma. But then we don't go much shallower than 20 meters often because that's where the kelp was. So often these sites were set up to be kind of the edge of the kelp and going a bit deeper because it's very hard to fish with hook and line in the kelp. You just get tangled in things. So most of our sampling has always been in places where it was sort of just beyond where the kelp was. But certainly there's going to be impacts when you lose, if the kelp's harboring lots of fish that tend to occasionally leave, right? You'll see some of those impacts. Carrying on, we do have 3 more years of data. We looked at those plots just recently. They look pretty much the same, right? It's still going up. There's a bit of variability in the past couple years, but it's kind of still doing that. I think we got information on kelp canopy biomass. That was another metric we put into some of those statistical models to try to explain the difference in response. It didn't come out as a strong predictor in this case, but it certainly is, I think, in those shallower depths where it's really present. We're often starting to get down into some species that I think it's the rock that's more critical for them, right, than necessarily the presence of kelp itself.
Thank you.
2 more stickers left after this.
Oh no. So I was just wondering about the examples you did find of fish being more abundant outside the MPAs than inside. Was it a consistent species that you were finding more abundant outside? Is it anything to do with taking advantage of there being less fish, other species around potentially?
That's a good question. Yeah, I mean, the ones that did, certainly the black rock fish, right, were more abundant outside, and a lot of that we think was probably due to kind of their mobility and things, and maybe a particular site that they really like. Another fish that was more abundant outside the MPAs was in Southern California, was California scorpionfish, which I always found to be kind of interesting. Like, why would that be? And they're another species. Typically, they don't move that much, but they do get together in these large spawning aggregations and move a lot. So it's another species that does a lot, at certain points of the year, does these large migrations. So it may have to do with kind of that mobility component to an extent. Yeah, and then a bunch of the other species that were more abundant outside were often ones that we didn't encounter that often, and so I think that's part of just the sampling error leading to it. You might expect, though, that if we sampled more of the non-target species, more of the lower trophic level prey species, that they could be more abundant outside where there's fewer predators. So, but we would need to have information on kind of the full complement of the community to really address that.
Yeah, I'm not sure how you fish or if you use bait, but I was wondering if there are different ways of fishing or different types of bait that might attract some species more than other and maybe influence your diversity estimates.
Great question. Yeah, I glossed over that. So the methodology, so each of the institutions uses 3 different gear types for fishing. Across all the institutions, the standard gear type is a shrimp fly with squid bait. And for us, we also use shrimp flies without bait, and then we use a jig, or we call a lingcod bar. So we have 3 different gears that are being, because they potentially will catch different types of fish. When I fish, I just catch the bottom, and that's about it. But what we've found, interestingly, is that those 3 types of gear, 1/3 of the fish are caught on each type. So we're catching the same numbers of fish regardless of the gear type. There are some differences in the composition, like you're more likely to catch lingcod, right, cabezon, some of the other larger rockfish on that bigger jig, the lingcod bar. So there's a bit of a difference in species composition, but in general the catch rates are basically the same, which is really interesting. Then in Southern California, instead of using the jigs, they use swim baits, which looks like a little rubber fish that swims around. And so there's a bit of a difference as well. They use circle hooks instead of the shrimp flies. So there's a little bit of a difference place to place depending on the types of species and the type of gear that fishermen use, most often in those regions. Yeah, great question. One more. Well, one more sticker. You can still ask more questions, but
I don't need a sticker, thank you. I've got one.
yeah, well, you're part of the program. Yeah. You can answer your own question. You're the one analyzing these data.
Yeah. Well, this is asking about another piece of data that we can throw into the pot. So this is kind of going off of Ivano's question about connectivity. Do you know what that group, you said it was Pete Raimondi and others that were working on that?
Yep.
What kind of spatial resolution they're working with, and you know, can you assign, for example, a connectivity score at the level, like what's the lowest level of that sort of thing that you could assign?
So, I mean, those ROMS models release particles, you know, into the ocean and track, you know, very accurately the position of all those particles, but where they land, sort of the coast is broken down into these also cells. There's maybe, like the whole coast of California, there's maybe 150 of them. They're probably 20, 30 kilometers long. And so they're tracking kind of where the particles are going within each of those cells, right? And so those connectivity matrices then are based on, you know, the connections between each of those sort of chunks of the coast, you know, 20 to 30 kilometers of coastline. Yeah, so that's the resolution that we would have to get at this, right? And some fully encompass the MPA, some maybe part of the MPA, right?
Right.
Because
Yeah.
so there's some challenge there in trying to figure out some of that, but yeah.
But yeah, I mean, that seems like there's some potential to add that as a covariate.
Yeah, I think so for sure. Ailey, last one. Last sticker.
I'm very much not a fish person, so pardon if this question just maybe isn't it. But you were targeting different species of fish, but because you're across northern, central, and southern California, are these all in their geographic range? Or like some of the fish like at the edge of their limits in Northern California, so you maybe just wouldn't catch them as much? Like how was that accounted for in like species distribution and like their optimal home range?
Yeah, I mean, so the distributions are changing, and we're catching a different set of species, right, in each of those regions. And in each one of those regions, you're occasionally catching ones that are much more common in the other ones. So like here in Central California, you know, mostly we're catching rockfish and lingcod and things, but occasionally we'll catch California sheephead, which have much more southern distribution, ocean whitefish as well. And those are species that they've kind of shifted a bit more north in response to the heat wave. And so you'll see that kind of in each of the regions. There's like some new species that are starting to show up a bit more frequently.
Gotcha.
And how do you deal with it? Well, usually the numbers are pretty low, and so often it's more of like a note of interest than anything. But it takes a while then for them to become abundant enough that you can actually then use the data, you know, in these analyses. So yeah, but it's an important question because we're seeing these northern range shifts in a lot of species, you know, along the coast. So and this program can be used to help, you know, detect some of those shifts.
The shifts in phenology.
Yeah. Yeah. Oh, one more question. No, we can get you one.
I was curious if you've compared your catch data to data maybe from other sources, like, I don't know, maybe sonar or satellite imagery for measuring abundance in a different way?
So we've compared it mainly to like the kelp forest scuba monitoring, as well as some like the video lander stuff. And so there was a recent paper that the group in Santa Barbara published that's part of CCFRP. It also works with Rick on the deep water and does the kelp forest monitoring, where they compared in the same locations. What are you seeing when you do scuba surveys, when you do our fishing surveys, right, or these sort of video lander surveys? And you know, in general, they were pretty comparable, right? Like where you saw positive responses, you tended to see it across programs, but they also sampled a different complement of species. So it's like they're showing generally similar results, but they're also complementary because they're not all sampling the same species. Even the divers versus just the video cameras, because the presence of divers can affect what fish tend to, some are attracted, some are repelled, and so yeah, the tool that you're using gives you a bit of a different picture. But luckily, like we still see MPA effects, like kind of regardless of the tool that's being used. Yeah.
Thanks.
All right. Thanks, everyone.